A quick note before we continue. I want to remind you guys that the reason we study cell biology is because this is how we target medicines. This is what medicine works on. It's at the cellular level. Often it's specific to an organelle. For example, the drug colchicine works on microtubule formation. There are such things as potassium channel blockers. There's calcium channel blockers. These are all medications that are targeting specific features of the cell. So our understanding of the cell is our basis for medicine. This is why it's so fundamental to your studies, to invest a little bit extra time on your cell biology. Okay, so on into the cytoplasm and contents thereof. So cytosol is also known as cytoplasm. You can also call it intracellular fluid, the intracellular environment. There are a lot of different names for it that you might run into while you're looking things up. So it's a viscous syrup-like fluid containing many different dissolved substances. It's got ions, nutrients, proteins, carbohydrates, lipids, and other small molecules. So everything that you are made up entirely of cells. It's all based on proteins, carbohydrates, and lipids. This may be familiar from a little thing you might have heard of called nutrition. You need every single one of those. You can't just stop eating carbohydrates. You can't stop eating lipids. You can't stop eating proteins. You need all three to build up your cells. Other small molecules are going to be things like vitamins and minerals. That's also nutritional. So the nutrition that you consume builds your cells, which builds you. So there's your cytoplasm, and I've got a coloring page for you online as well. I believe I've referred to that already. So it's going to end up looking like this. You're going to want to label everything on that coloring page, though. It's more like this, where I haven't told you what everything is. So inclusions. Inclusions are just going to be something that is in the cytosol, included, literally. So melanin is an example of an inclusion. Melanin is a protein. It's made by something called a type of cell called a melanocyte. It is what gives your skin, and if you have brown eyes, the pigment, and it's an inclusion. It's a protein that gets deposited into those cells in that case, and because it is included in that cells, it becomes a feature of that cell. Glycogen is a long chain of sugars. You'll find it a lot in the liver and skeletal muscle. This is going to be how we store sugars that we get from our diet to be a quick source of energy when we need to create a lot of ATP. So I don't think we're doing much with cellular respiration, but remember, your cellular respiration is breaking down a glucose molecule into ATP, that molecule that we can use for energy in the cell. Organelles can be membrane bound or non-membrane bound. Most of the ones we focus on are membrane bound. They mean little organs. They all perform a different function, and the type and number of organelles within a cell is a reflection of the cell's function. So again, remember, this is prototypical cell. It can be arranged any way that we need these organelles to be arranged to do a specific function. If we need contractibility, like in a muscle cell, we're going to have a whole lot of cytoskeleton lined up in a way that can shorten and lengthen, for example. So your membrane-bound organelles, here's your list, and I believe on your worksheet I've given you pictures of most of these to describe and associate their descriptions with the visual aspect of them, with the way they look and their names. So your endoplasmic reticulum, the Golgi Apparatus, lysosomes, peroxisomes, and mitochondria are all examples of membrane-bound organelles that we will cover. Starting with your endoplasmic reticulum. So remember, your nucleus is its own thing. You could say it's located within the cytoplasm, but it is our control center of the cell. It's the brain of the cell. It's in the middle. We don't count it officially as part of the cytoplasm. Our three parts of the cell are plasma membrane, nucleus, and cytosol. So the nucleus is not officially part of the cytosol as a category. So our first thing out from the nucleus, the first feature located superficially, let's use your directional terms, to the nucleus, is the rough endoplasmic reticulum, followed by smooth endoplasmic reticulum. So maybe you remember from high school, the thing that makes rough endoplasmic reticulum rough is the ribosomes. This is a nice transmission electron microscopy image of rough endoplasmic reticulum. There's our lipid bilayer, phospholipid bilayer. This is the membrane. This is a membrane-bound organelle. And all of these little circles, those are all ribosomes. So the rough endoplasmic reticulum is studded with ribosomes. The smooth endoplasmic reticulum is not. It's going to have a job. Its job, those ribosomes, you remember what ribosomes do? What ribosomes do is they synthesize proteins. They take the information that was transferred from DNA. It was DNA to mRNA. They take the mRNA, that's the instructions for a protein. tRNA comes in with an amino acid, adds an amino acid to the ribosome. The ribosome polymerizes amino acids into a long chain and creates a protein. So, therefore, the part that's important for this is that rough endoplasmic reticulum is mainly for protein synthesis. And the mechanism by which they create proteins is the ribosome. The smooth endoplasmic reticulum, and again, so here's what it looks like in 3 dimensions. Let's step back for just a minute. We've done a sort of transverse section through this cell's nucleus and endoplasmic reticulum to see the relationship. But in 3 dimensions, you've got these cisternae, essentially. So remember, this is cut in half. They would be complete. They would be extending up in the superior direction, going up and enclosing in 3 dimensions. But we almost always visualize it cut open like this so that we can visualize the internal portions of the cisternae. Smooth endoplasmic reticulum is also metabolism. You could argue that this rough endoplasmic reticulum was metabolism as well. That was building up, which is defined as anabolism. Metabolism of carbohydrates and lipids is going to occur on the smooth endoplasmic reticulum. So big picture, proteins, fats, and carbohydrates, going back to that idea of nutrition and the things you consume, building the things in your cell. So this is how we modify things in the smooth endoplasmic reticulum. We build them up, we break them down in the endoplasmic reticulum. The smooth endoplasmic reticulum is also special because it detoxifies some drugs. It's more metabolism. We detoxify drugs by modifying them. We might add something to them or remove something from them. And we change those drugs into a form that our body can get rid of or store safely if we can't physically get rid of it. So for example, alcohol. I've actually heard contradictory things about where alcohol is detoxified. Some people say smooth endoplasmic reticulum. I've also heard peroxisomes as being the location for alcohol detoxification. For our purposes, let's assume the smooth endoplasmic reticulum is our site of detoxification of alcohol. This is definitely not something you need to memorize, but for those of you who are wondering how we actually detoxify something as a metabolic pathway, essentially this is what's happening in your smooth endoplasmic reticulum is we are taking ethanol, the alcohol that you drank, and adding alcohol dehydrogenase. That's going to be an enzyme. If it ends in -ASE, that is going to typically be an enzyme that is going to modify a substance into something else. In this case, we modify ethanol into acetaldehyde. And then at another point, we modify it into acetate, which is harmless to our body. And then we can get rid of this. We can urinate this out, essentially. So pretty cool. Smooth endoplasmic reticulum, especially in the liver is where this is taking place. Your liver cells are going to have plenty of endoplasmic reticulum. And we talked about rough ER being all about synthesis, transport, or storage for proteins. Those proteins are going to be anything, right? They might be an enzyme, they might be secretory products such as neurotransmitters. For example, they might be transmembrane proteins or integral proteins. They might be creating lysosomes. We're going to talk about lysosomes in just a minute. So whenever you need something functional for the cell, it's probably coming from the rough endoplasmic reticulum. So the Golgi apparatus is also all about proteins. In this case, we're going to package and modify those proteins. We're also going to transport them from our endoplasmic reticulum to the periphery of the cell, to that plasma membrane in many cases. So again, Golgi apparatus, transport, modification, and storage of proteins. Oh, there it is. Modification, sorting, packaging, proteins, and a bit of lipids. Again, you have cisternae. Again, we can consider this as something that we've cut through in a transverse section, but in reality, it is in 3 dimensions. And then you have a sort of arrangement. You have a cis face and a trans face. The receiving region is the cis face. The shipping region is the trans face. Consider the roots of these words. Cis means same, trans means opposite or across. This is a phrase you'll see a lot in chemistry in general. So that helps you make sense of its positioning. So the cis face facing the rough endoplasmic reticulum, the trans face facing the plasma membrane. So again, we have to talk about vesicles. I believe this has already come up. A vesicle is going to be a bit of phospholipid bilayer in a sphere, and inside that sphere is something, in this case, protein. So that vesicle in this case is a transport vesicle, transporting protein. These vesicles will tend to be pinched off from one membrane-bound organelle and passed on to the next in sequence. So we go from the rough endoplasmic reticulum, we might have to go through the smooth endoplasmic reticulum, and then to the Golgi apparatus. It's all made out of plasma membrane. That's essentially what it is. So that whole, very much like the endocytosis and exocytosis, we have the merging and then the pinching off of plasma membrane as things are transported along from one part of the cell to the other. Like this. So here's our endoplasmic reticulum. A vesicle containing proteins pinches off. The plasma membrane merges with the cisternae plasma membrane of the Golgi apparatus. And then when it reaches this other side, there are probably enzymes in here that are going to modify it. Then it reaches the other side. And yet again, a piece of endoplasmic, sorry, not endoplasmic reticulum, a piece of plasma membrane pinches off and creates a vesicle on this side, which perhaps will skip one part of the cisternae or not. Perhaps we're going to go through every one. And then you get to the shipping region, and at this point, a final vesicle pinches off from the plasma membrane of this cisternae and joins with the plasma membrane on the outside of the cell, the actual plasma membrane, and exocytosis occurs in this case. In this case, these are membrane-bound proteins. They simply bind into the plasma membrane of the vesicle, and when they get to the plasma membrane of the cell, then all that has to do, the plasma membrane just has to merge with the rest of the plasma membrane, and the fluid nature of the plasma membrane allows for movement of those integral proteins into various parts of the outside of the cell. Cool, right? It's delightful. All right, so a lysosome is a very specific kind of vesicle. So it's still bound in plasma membrane. It's still that phospholipid bilayer. In this case, though, it's going to contain digestive enzymes. We talked about this a little bit earlier when we talked about endocytosis and phagocytosis. So lys means to cut. So this is going to be a sphere, a some, that contains digestive materials that are there to specifically digest material that enters via endocytosis, or remove worn out or damaged organelles, or be part of self-destruct sequences, something called apoptosis that you're going to hear quite a bit about later on. Okay, they're generated by the Golgi apparatus. Of course they are, because they contain enzymes, which are proteins, which are modified and packaged in the Golgi apparatus. Yeah, we got this. Autophagy means self-eating. Autolysis means self-cutting. Peroxisome is specifically to detoxify. Specifically, one thing it does, convert hydrogen peroxide to water. It's going to have a sort of crystalline core that's going to help with this process. It's going to also break down fatty acid molecules, for example. So along with the smooth endoplasmic reticulum, it's part of our detoxification process. Visually, it's hard to designate what a peroxisome is going to actually look like. When I draw a peroxisome on the board, usually trying to draw something similar with a shape, I draw a kind of like diamondy thing in the middle to symbolize our sort of crystalline core. That's about as close as I can get with our PowerPoint tools. It'd be better if I had a blackboard. Okay, it uses oxygen and the enzyme catalase to detoxify harmful molecules taken into the cell. The enzyme catalase is a cool one. I used to do an entire lab around the catalase enzyme. Hydrogen peroxide, know about that. Hydrogen peroxide is a metabolic byproduct. It's something our cells produce. It's actually harmful to the cell. It's an oxidizer, which means it steals electrons. Not really a good thing for your cells. So we actually need to break down hydrogen peroxide into water and oxygen in order to clear it away. You don't necessarily need to learn that, but it's cool. Okay, mitochondria. Mitochondria is the powerhouse of the cell. It's actually kind of a stereotype that that's one of the only things you ever learn in college is what mitochondria are. It's in a lot of memes for some reason. Nevertheless, it is the powerhouse of the cell. This is the location for aerobic cellular respiration. This is where we create the bulk of our ATP molecules used in all of these cellular processes. Anything that requires energy, such as moving ions against a gradient, for example, is going to utilize ATP created by mitochondria. They have a double membrane, an inner membrane folded into cristae. The fluid inside of this is called the matrix. High yield ATP creation, and you will find more mitochondria in cells that require more energy compared to cells that do not require as much energy. Non-membrane bound organelles include ribosomes. There are ribosomes on the endoplasmic reticulum. There are also free ribosomes. The cytoskeleton, no plasma membrane there. Centrosomes and centrioles. These are part of your mitosis process. Cilia and Flagella and Microvilli. And we're going to specify the function of this versus this. They get confused very frequently. Let's keep them separated. So ribosomes are made out of protein and RNA. They are actually called rRNA when you go to study transcription and translation. And you will see transcription and translation at some point. I just did this lecture for pathophysiology, actually. So free ribosomes float unattached within the cytosol. Fixed ribosomes are attached to the rough endoplasmic reticulum. There's two subunits, small and large, and when they come together, they synthesize proteins utilizing information copied from DNA. Cytoskeleton, you have three types of cytoskeleton: microfilaments, microtubules, and we honestly don't hear as much about intermediate filaments, but they're there. Microtubules are made up of tubulin. Microfilaments are made up of actin. You will need to know all about actin later. Microtubules are relevant to our mitosis discussion in just a bit. So the centrosome, again, it's made up of microtubules, and it's a special organelle that is only present for mitosis, for the copying, for division of a cell from one cell into two genetically identical daughter cells. So microfilaments, that's our actin. Microtubules, that's our tubulin intermediate. Microfilaments are all about cell shape. Breaking these down and building them up can change our cell shape, especially if that cell needs to move around. It'll develop something called a foot process that is made out of microfilament. It's a helical strand. It participates in muscle contraction and cell division. So our muscle contraction from microfilaments, you're going to need to know all about something called actin and myosin that will be found in this banded pattern here. Later this semester, we will talk all about it. Intermediate filament protein composition varies, provide structural support and stabilize junction between opposed cells, as in cells that are right next to each other, not opposed, they're not against each other, they are apposed, they are adjacent. Microtubules radiate from the centrosome, composed of tubulin protein, hollow tubes. They fix organelles in place, they maintain cell shape and rigidity, and direct movement of organelles into the cell. They are also what creates motility in cilia and flagella. So I mentioned where is it? Nope, farther back. Here, we're going to separate the functions of cilia and flagella and microvilli. So we just said that cilia and flagella have movement. This is the key aspect. Cilia and flagella have movement because of the structure of microtubules. We'll get to microvilli in a bit. And they move chromosomes during cell division, so mitosis. Again, the centrosome and centrioles. Centriole is going to guide cell division, and it's made out of microtubules in a very specific arrangement. All right, cilia and flagella. Again, this is movement. Cilia and flagella are about movement. The difference between cilia and flagella is that a cell will have many, many, many cilia. They are going to wave. The cell stays in place. The stuff on top of the cell is going to move because of the movement of the cilia. So in this case, what this is demonstrating is, I believe this is going to be your bronchial tree. This is the trachea. This is the air tube to your lungs. Has a layer of mucus. If you inhale some carbon pollution, some particles, some dust, it's going to land in this layer of mucus, and the cilia are going to brush it in a superior direction so that instead of inhaling this particle, you're going to cough up this particle, which is very good for your lungs. You don't want to be inhaling a whole bunch of carbon pollution, a bunch of bacteria or viruses. You want it to get trapped in that layer of mucus, and you want that cilia to be able to push it away from where it can hurt you. There's only one cell in human bodies that has a flagellum. It's very common in nature and other organisms, especially small multicellular organisms. But in humans, the only thing with a flagellum is the sperm. So even only males of our species have any cell with a flagellum. Flagellum is singular, and its function is to move the cell. So the flagellum is the structure that allows the sperm to be motile and to swim up the reproductive tract and search out a fertilized egg. So again, cilia and flagella are both about movement. Cilia are numerous. They move the thing on the outside. Flagellum are singular. A flagella is singular. Flagella is plural. Flagellum is singular. And it moves the cell. Microvilli, by contrast, they look similar, right? It's cellular extensions. This is extensions of the plasma membrane. There's no microtubule structure. There's no power stroke. It does not move. It's there to increase surface area. We're going to see this concept a lot. I'm going to ask this question a lot, and you can look like a rock star by answering it. Why do we have invaginations? Why do we have folds? Why do we have microvilli? To increase surface area for something to take place. So one large purpose of microvilli is absorption. You're going to find a lot of microvilli in the small intestine, where we are absorbing most of the nutrients that we got from food into the body. So it's there to increase surface area. It does not move the cell, it does not move stuff on top of the cell. It increases surface area onto which reactions can take place, absorption can occur, things like that. So if you want to take a moment and name two differences between cilia and flagella, and even better, add another difference between cilia, flagella, and microvilli. That's a good concept check. See if you're understanding this. All right, our last organelle is, or sorry, not organelle, our last part of the cell. We've got three parts of the cell: nucleus, plasma membrane, and cytosol. The nucleus is our last one, but it's also very, very important because it's the brain of the cell. It's our control center of the cell. It contains DNA and a nucleolus. So people always forget what the nucleolus is. The nucleolus is the structure. It's the structure that assembles ribosomes. I mentioned earlier that ribosomes are made of protein and RNA. So what is in the rest of the nucleus is DNA. When DNA gets copied onto something that can separate from the DNA, we call that RNA. It's a single strand. And then we can use that RNA to create proteins. The ribosomes are the rRNA that actually, sorry, the ribosomes, they're being assembled in here. This is, out here is chromatin, this is genetic information. Ignore where my cursor was just then. So rRNA is ribosomes. They are assembled here in the nucleolus. mRNA is copied from DNA, taken out to the cytosol. tRNA brings amino acids to the ribosome for assembly. So that's what the nucleolus is the site of ribosome assembly. This whole thing is the nucleus. Half the time when I ask students to label the nucleus on things like this, they just label the nucleolus by accident because they don't recognize that this environment here is the cytosol, not this. This is DNA. This is the inside of the nucleus. When the nucleus is in its normal state, the DNA is loose. This is called chromatin. It's only when we undergo mitosis that the DNA condenses into chromosomes, which we'll get to later. The nucleus has this double membrane here, membrane there, and it's spotted with nuclear pores. These nuclear pores are going to be very selective about what goes into or out of the nucleus. Some things that can leave the nucleus, mRNA, messenger RNA. Some things that can bypass the system entirely are steroid hormones. So estrogen can go bloop through that, doesn't even have to care about nuclear pores. So the nucleus is made of the nuclear envelope, has the two phospholipid bilayers, nucleoli, DNA, which is either in a state of chromatin or chromosomes. Both chromatin and chromosomes are forms of DNA. I think we have covered all of that. The nuclear envelope controls entry and exit of molecules from the nucleus to the cytoplasm. Outer membrane is continuous with the endoplasmic reticulum. This part of the membrane actually folds off and becomes our rough and then smooth endoplasmic reticulum. They are continuous structures even though we consider them different parts of the cell. And again, nucleoli composed of RNA, enzymes, and various proteins. Its job is to construct the components of the ribosome. So differentiate again for yourself. Take a second, write it down. What does the nucleus contain? Sorry, that's spelled wrong. Having a George W. Bush moment. Nucleus. And what does the nucleolus? Wow, I could not spell while I was writing this. Nucleolus contain nucleus. It's nucleus, not nucleu. Okay, DNA, chromosome, chromosome. Okay, nucleus houses deoxyribonucleic acid, or DNA. When it's unwound, it's chromatin. When it's wound up tightly, it's chromosomes. Why do we need these two different stages? When it's chromatin, when it's unwound, we can copy it. The enzymes that copy it can't reach every individual part of the DNA strand when it's bound up into chromosomes. We need chromosomes for mitosis. DNA structure looks like this. It's a DNA double helix. It only goes in this one direction. Half the time in pop culture, you'll see a DNA double helix winding in the opposite direction. A lot of pop culture is guilty of it. If you learn which direction is correct, you will be pissed off for the rest of your life every time you see it going the wrong way. It's fun. So in its loose phase, in chromatin, it looks pretty much like this. But then you can get this to wind around something called histones. And then those structures that are wound around histones wind around themselves, which wind around themselves, which wind around themselves, until you have a 6-foot length of information condensing down into a single chromosome. And guess what? You have 23 pairs of chromosomes, 46 chromosomes total in every single cell in your body that maintains a nucleus. It's a crazy amount of information. Okay, I'm going to hit pause and do mitosis as our last part separately.